In this video, I hope to provide a solid understanding of something that is fundamental to neuroscience, membrane potential. To understand membrane potential, it is first prudent to obtain a grasp of the basic physics of electricity. Here on the left is an electric field, which in simple terms can be defined as the force that surrounds electrically charged entities. In this case, a positive charge at point A exerts a force, and if we were to place a positive charge near point A force would push it toward the relatively negatively charged location B. In this case, when a free positive charge Q is accelerated by an electric field, it is given kinetic energy as it moves from point A to point B. This process is analogous to an object being accelerated by a gravitational field. For example, when an object is dropped from a high point, it gains kinetic energy as it moves downward due to the force of gravity. For electricity, it is as if the charge is going down an electrical hill where its electric potential energy is converted to kinetic energy. In this instance, work can be described by work is equal to the negative change in potential energy. That is, the work done to accelerate a positive charge from rest is positive and results from a loss in potential energy or a negative delta potential energy. Therefore, there must be a negative sign in front of delta potential energy to make work positive. Using this idea, potential energy can be found at any point by taking one point as a reference and calculating the work needed to move a charge to some other arbitrary point. Electric potential, or simply potential as indicated by V, is defined as the potential energy per unit charge. The change in potential energy, shown as delta potential energy, is crucial, and we are concerned with the difference in potential, or potential difference, abbreviated delta V, between 2 points B and A. The potential difference between points A and B, as indicated by V sub B minus V sub A, is thus defined to be the change in potential energy of a charge Q moved from point A to point B divided by the charge. Units of potential difference are joules per coulomb, given the name volt. Joule, of course, is the SI unit for energy, being a product of force and displacement, having the base units kilogram meter squared per second squared, while C, or coulomb, is the SI unit for charge. The familiar term voltage is the common name for potential difference. Electric potential, or voltage, across biological membranes is established by membrane proteins that facilitate the movement of ions. This results in a balance between the concentration of these ions and the electric charge across the membrane. This occurs by virtue of the energy-dependent processes of, for example, sodium-potassium ATPases, during which 3 sodium ions are pumped out of the cell, while 2 potassium ions are pumped into the cell. The resulting balance, as mentioned prior, establishes an electrochemical gradient. As shown, the outcome is a greater concentration of sodium outside the cell relative to inside, and a greater potassium concentration inside the cell compared to out. Millimolar concentrations of these ions in the extracellular and intracellular spaces of a typical neuron are shown in this table. As mentioned, the concentration of sodium outside the cell is much greater than inside the cell, being 145 millimoles per liter in the extracellular environment, while the intracellular concentration ranges from 5 to 15 millimoles per liter. By contrast, potassium exhibits a much higher intracellular concentration relative to extracellular, with 140 millimolar concentration on the inside of the cell and a 5 millimolar concentration outside the cell. The resting membrane potential across cell membranes is largely established by potassium ion concentration, and in the majority of neurons is approximately equal to negative 70 to negative 75 millivolts. Recall that voltage is essentially the difference in electric potential energy between 2 points. In this case, the electric potential energy is measured inside the cell relative to outside the cell. Since the extracellular environment is more positively charged compared to the inside, there exists a difference in electric potential energy between these 2 points. This can be understood as the inside has 75 millivolts of electric potential energy less than the outside. This is because voltage is not a measurement of energy itself, but instead is concerned with the difference in electric potential between 2 points, as stated prior. In this case, if a positive charge was placed outside the cell, a force would be produced to move that charge away from the positively charged exterior in accordance with the common understanding that like charges repel and opposite charges attract. Placing a positive charge on the inside of the cell would similarly induce movement of the charge away from the positively charged interior, but the lower potential energy in this environment would result in overall lower kinetic energy as the charge is moved. This lower kinetic energy is thus less than would result in the exterior as the environment has a larger magnitude positive charge. That is, the extracellular environment has a larger magnitude positive charge. This can be further delineated by referring to the equations and relationships shown here. If you'll recall, energy is conserved, and thus the change in potential energy will equal the change in kinetic energy. Algebraically manipulating this relationship given by voltage is equal to the change in potential energy per unit charge, we see that potential energy is equal to the charge times the potential difference. Thus, the resulting kinetic energy that would occur from a constant charge Q, such as an arbitrary positive charge shown here, moving in these two different environments, would have greater kinetic energy in a situation where voltage or electric potential is larger. Based on this pragmatic thought process, it seems intuitive that the interior therefore has less electrical potential energy than the exterior. Referring back to the negative value for voltage, if we look back at the formula for voltage, we know that this is a situation in which the potential is greater at point A compared to point B, thus producing a higher value at point A being subtracted from a lower value at point B. This results in a negative output and therefore provides another avenue to describe the reason for the resting membrane potential reading negative. Now to take a look at membrane potential as it relates to the classic graph of an action potential firing. Since we now have a clear understanding of voltage and what this really means, this graph becomes more intuitive. It is easy to memorize that action potential results from depolarization of the membrane as characterized by a rise in the voltage value, but what does this mean in terms of what we've just described? Essentially, as the sodium ions influx through, for instance, ligand-gated sodium channels, the interior of the cell becomes more positively charged relative to the outside. This means that the difference in electric potential energy between the exterior and the interior diminishes, resulting in a lower negative millivolt value. That is, the potential difference approaches zero. Next, threshold potential is reached and voltage-gated channels change conformation and open in response to the altered constituent amino acid electrostatic interaction with the environment. To briefly expound on this, recall that certain amino acids have charges on their side chains which are sensitive to the environment, such as pH and any electrostatic forces. Normally, protein conformation is a balance between deterministic and thermal forces that dictate the structure in line with thermodynamics, with proteins being free energy minimizers and, by extension, entropy maximizers. Therefore, any change in the environment, such as the interaction between the amino acids and their ever-changing environment, necessitates changes in the conformation of proteins to uphold maximum entropy. The result, in this case, is the opening of the voltage-gated channels, and this promotes a larger influx of sodium ions, leading to the depolarization of the membrane, wherein the interior of the cell becomes more positively charged relative to the exterior, providing it with greater electric potential energy if a positive charge were to be placed within this environment. This outcome is what we know as an action potential.